Hello everyone, today I'm going to talk about protein diol synthesis. Now, in order to understand this procedure, first we need to identify the constituent of a protein. Although we may have different shapes and different size proteins, but the basic procedure of protein is a chain of amino acid. Just like this necklace is made of pearls, and the amino acid is just like the pearls in the protein. Next, we will see the structure of a DNA. As you can see in this picture, DNA has two strings, and these two strings can pair with each other, just like a zip. Next, we will see the structure of mRNA. mRNA is also like a part of a zip, but it's only one side of a zip compared with DNA. Okay, tRNA. In order to understand what is a tRNA, we need to understand 3 points. The first point is tRNA is also come from like a portion of a zip. And the second point is if mRNA is A side of a zip, tRNA will be B side of the zip, which means mRNA and tRNA can combine with each other. And the third point is tRNA is connected with an amino acid, just like a portion of a zip connected with a pearl. Okay, furthermore, we need to learn the structure of a cell. A cell has two areas. The first area is the nucleus, and outside the nucleus is the cytoplasm. As we can see in this picture, DNA exists in nucleus, while tRNA exists in cytoplasm. Here is the general look. At this point, you may ask me, where is the mRNA? In fact, we don't have steady mRNAs. If the cell wants to use mRNA, the cell will synthesize it. After being used, mRNA will be digested by some enzymes. Okay, let's come to our point today. How can we get a protein? In order to get a protein, we need two basic procedures. The first procedure is called transcription. In other words, to synthesize mRNA. As mentioned before, we don't have steady mRNA. And in this part, this procedure happened in nucleus, and the DNA strings will depart with each other, and run sheet will add one by one based on one string of DNA. And also this run sheet will hand in hand together to make a chain of mRNA. After this chain has formed, mRNA will left DNA string, and DNA will recover its steady state. mRNA left DNA and the nuclei and will enter the cytoplasm. In this place, mRNA will encounter tRNA. tRNA can combine with mRNA. In other words, also we can know amino acid will be pulled together. Amino acid will also add one by one to make a chain, and they will hand in hand to make a steady structure. After amino acid chain has synthesized, this chain will left tRNA, and mRNA will be digested. Just left the chain of a protein like a necklace. After we have this chain, however, we still don't have mature proteins. After After this procedure, some other procedures also needed. Maybe to squeeze the chin or curl the chin, just like this necklace, to make the final condition or final structure of the protein. Okay, here's everything about today. I hope you can understand this basic procedure in biology area, as it's very important for your future studying.